Hello everyone, very good morning to you all. I think you all can hear me. Sorry for the delay for the few minutes. So I warmly welcome you all to the technical session on agricultural production and environment. This session has 6 presentations. So first, let me introduce the chairperson and the judges today. It is my privilege and pleasure to introduce the chairperson for this session, Professor Nimal Pereira, Professor Emeritus, Department of Animal Science, Faculty of Agriculture, Institute of Peradeniya. Thank you, sir, for being here today. And also we have 3 eminent judges today. I kindly request all the judges to turn on the video for a while so that the presenters can see you. First, Professor D. Cruz, Professor, Department of Aquaculture and Fisheries, Faculty of Livestock Fisheries and Nutrition, Biome University of Sri Lanka. Then Dr. Nalaka Gikiyanage, Senior Lecturer, Faculty of Agriculture, Rajaratnam University of Sri Lanka. Then Professor T. Amarasekara, Director, University College of Batangala, Aligoda. Thank you, sirs, for your valuable time and being here today. So let's move to the presentations. We now cordially invite Professor Nimal Pereira, sir, to chair the session. Over to you, sir. Thank you.
Good morning, everybody. I welcome you all for this session. So I would like to give a small instructions regarding the presentation to all the presenters. Your presentation time will be limited to 12 minutes, followed by a 3-minute discussion. Right? All your proceedings will be recorded. The presenter must be adhere to the time limits. And anybody who wants to raise a question after each session, you use the raise hand option and the question will be delivered to me to put it to the audience. Then soon after your question is attended, please lower the hand option so that we know that you have already been asked the question. Presenters must mute the microphones at the end of the discussion, right? Okay. So without taking much time, since we are a few minutes behind, let's start with the first presentation. The first presentation title is Population Parameters of Peneus semisculatus caught by pike net fishers from Kachai fishing ground of Jaffna estuary, Sri Lanka. The authors are N.N. Viturasha, N. Raghavan, and U. Edrising. The presenter is N.N. Viturasha. So I'll invite N.N. Viturasha to make your presentation.
Hello, ladies and gentlemen. I am Viturasha. Today I am going to present my research finding on population parameters of Peneus semisculatus caught by pike net fishers from the Kachai fishing ground of Jaffna estuary in Sri Lanka. This is the content of my presentation. First one is the introduction, objective, site selection, data collection, results and discussion, conclusion, finally the references. First one is the introduction. Jaffna estuary is the largest brackish water body which is located in the northern province of Sri Lanka, which lies between the Jaffna and the Kilinochchi district. The estuary is enriched with vast biodiversity of the aquatic biota, especially in shrimps. Shrimps made highest contribution to estuary landings, and it was higher than 25% total unloaded harvest from the Jaffna estuary. There are several ways used to operate harvesting for the shrimp. For example, triangular shape dragnet net, push net, brush file, straps, rubber dragnet, hook net, and handling. Only few studies have been carried out to investigate the different dimensions of the pike net fishery in the Jaffna lagoon. And the pike net is the major gear used to caught the shrimp in the Jaffna estuary. So that's why I select this topic for the investigate the pike net fishery in the Jaffna lagoon environment. The objective of my presentation is identify the shrimp diversity in the pike net fishery in the Cascade landing site from June 2019 to May 2020. And second objective is acquire the knowledge on population dynamics of Phineas semicircular species which are caught in the pike net fishery, which will be helped to manage the stock in the Jaffna estuary area. This is a site selection. I selected Cascade landing site of the Jaffna estuary, which is located near to the Savacheri DS division. This is the coordination, the latitude, longitude of the Cascade area. For the data collection, I collected data from June 2019 to May 2020. Randomly, I selected samples from the pike net fishes from Cascade landing site twice a month. Per month, 2 times I visited to the Cascade landing site and collect the samples and identify the particular samples by using the FAO guide book. And length and weight of the Phineas semicircular was recorded. And the 5 set 2 used for the data analysis part. For the results and discussion, First, I analyzed the morphometric characters of the strings. I measured the total length of the strings and the total weight of the strings. Separately, I measured for the male, female, and pooled one. I entered all the data into the 5 set and I get the table like this one. And I get the table like this one: mean length, minimum length, maximum length, mean weight, minimum weight, and the maximum weight. When we observe this table, we can find out the female has the highest total length and the body weight also female has highest one because the mean length is 11.71 and the mean weight is 11.03. The female showing the highest growth rate than the male one. Then secondly, I analyze the length-weight relationship of the particular species. Regression relationship between the total length and body weight of the female and male of Phineas semicircular species were determined separately. When we observe this table, you can find out here the R-squared value is higher in the female species than the male one. That means they have the stronger regression relationship between the total length and the body weight of the particular species. And the B value 2.9 for female and 3.1 for male. Which one is approximately similar to same for 3. So the both are showing the isometric growth. Isometric means when the weight is increasing, their length also increasing. They are showing the isometric growth. Then I analyze the growth parameters. Under the growth parameters, asymptotic length, growth curvature constant were estimated for males and females of Phineas semicircular separately. And I recorded. Female species saw the highest symptotic length than the male one. And the growth curvature constant are high in the male species than the female one. Then I analyze the recruitment pattern of the particular species. Recruitment means they are producing the next generation. They are releasing their eggs for their next generation. So recruitment pattern of the Phineas semicircular I analyzed. When you observe this graph, you can find out the recruitment period of the particular female semicircular is the October to January. The trainee season is the favorable for their breeding time. Then I analyze mortality rate and the exploitation rate of the particular species. Mortality, natural mortality, fishing mortality, exploitation rate are analyzed, and I summarized all the results in this table. When we observe this table, you can find out the male has the highest total mortality, natural mortality, fishing mortality, and exploitation rate than the female one. When you see the exploitation rate, the male has the higher than the female one, but that value is less than 0.5. That means the resources are underexploited in the environment. So we can more utilize the resources in the area. Finally, conclusion part. Estimation of the population parameters of fishes is the key factor to understand the life history trait of the target organism in order to manage the fishery resources, while the growth pattern, growth parameters, mortality, and recruitment patterns of fishes are the important parameters to understand the life trait history of fishes, and several fishery models could estimate these parameters. So these parameters are important to manage the stream resources in the particular environment. Those are the references of my presentation. Thank you.
Thank you very much, Ms.
Finding population
Vertusha.
parameters of Peninsula Misericordia scored by 5 net fishes from the
Thank you very much, Ms. Vertusha.
Thank you, sir.
Are there any questions from the participants? Dilip De Cruz raised a hand.
Yes, sir. So, here Versusa, very nice presentation. I would like to ask one question from you. Here you have estimated, can you hear me? Am I audible? Right.
Yes.
Yeah. So here you have estimated the population parameters. So most of these population parameters are estimated for the entire population, because these are very important for the managerial decisions. So here, when you are estimating this, what you have selected the sample only from the phycinate.
Yes,
so do you think then these estimated population parameters are accurate and that those can be used for the managing the fishery?
I only have the 5 net fishes. I collect samples, more than 500 samples I collected for the research study. I think I have to analyze entire population. The other gears also practice in that area. No, I have to improve my research study, because I only analyze the 5 net fishes. No, I have to analyze the other cast net and other motorized boat, other fishing method also I have to analyze. But in the my main objective is analyze the 5 net fishes. 5 net fishes caught by the stream diversity. That's why I select only the 5 net gears. Approximately, is it okay? But I have to further analyze the other gears also.
Yes, exactly, because you know the phycinate is set in the estuary mouth, and stream migrate in between the estuary and also the coastal area. So you have selected the bias sample. And the other thing is, if you want to analyze the diversity and distribution of phycinate, you could have done that thing, but here you have estimated some of the population parameters, which need to be done by considering the entire population. So when you are publishing, it would be better if you can omit that section and go ahead with that.
Yes, sir.
Thank you.
Okay, thank you, sir.
Thanks.
Thank you very much, Dr. Cruz. The next question is for Dr. Nalaka.
Thank you, sir. Am I audible? I hope so.
Yes, you are audible.
Yes.
Thank you. Thank you. Thank you. The nice presentation we really enjoyed, and apparently it's a lot of data. I have 2 suggestions for actually to improve on your presentation, and maybe if you're writing a full-length article. One thing actually, your objectives did not certainly reflect what you did. The first one, if I recall well, the first one was about diversity. When we identifying the diversity, the only comparison as far as I concern, you presented is the female and male comparison. When it comes to diversity within a species, that's not actually the case. Also, if you presented maybe some method comparison of various methods, as you pointed out in the previous question, there would be appropriate, but a more appropriate objective would be you to evaluate the population parameters, that what really you have done. Also, when it comes to the conclusion, your conclusion was rather general in my opinion, and you did not really connect your conclusion to your results. And whatever the things you mentioned was the kind of benefits that you can do from generally you can do from these analysis. It would be better if you can relate your findings to the conclusion, so that will be more appropriate for a general reader. Thank you.
Yes, sir. Thank you, sir.
Thank you, Dr. Nalaka. Any more questions? Looks like there are no questions. I'd like to make a small announcement. I would like to have all the participants online until we complete the sessions. Number two, when the presenter is subjected to questioning, clarifications, please keep your video on. Okay, let's move into the second presentation. The title of the presentation is Effect of Elevated Temperature and Carbon Dioxide on Biomass and Sucrose Accumulation of Selected Sugarcane Genotypes. The authors are A.L.C. De Silva, W.A.J.M.D. Costa, and L.D.B. Suryagoda. The presenter will be A.L.C. De Silva. This is for you. Okay, you can start now.
Yes, and good morning, yes, sirs and madam. I am Chandrajit, would like to present the findings of our research experiment on the title of Effect of Elevated Temperature and Carbon Dioxide on Biomass and Sucrose Accumulation of Selected Sugarcane Genotypes. In this experiment, we evaluated the impact of climate change on sugarcane. Therefore, as the introduction to the presentation, in person, let's see how climate is changed and impact to the crop production. As shown in the figure, global temperature anomalies and carbon dioxide concentration in the air increasing continuously due to human and natural activities. It is leading to increased global warming and to change climate. Changing climate is a challenge in many aspects of crop production. However, very few studies have been done to evaluate the impact of especially elevation of temperature on cereal crops. As example, in sugarcane, only 5 experiments have been done to evaluate the impact of elevation of carbon dioxide, and only 2 experiments have been done to evaluate the impact of elevation of temperature, and they have shown that elevation of carbon dioxide concentration increases biomass production, and also in elevation of temperature up to 33 degrees centigrade increases biomass accumulation, but elevation of temperature beyond 33 degrees centigrade decreases biomass accumulation, and no one studies the impact of elevation of temperature beyond 35 degrees centigrade, and also no one studied the impact of even elevation of 33 degrees centigrade on sucrose accumulation of sugarcane. Therefore, dear sirs and madam, the objectives of our study were to determine the the responses of biomass and sucrose accumulation of sugarcane to elevated carbon dioxide and temperature, both individually and together, and to determine the genotypic variation of these responses in sugarcane under the range of elevation of carbon dioxide up to 750 ppm and elevation of temperature up to 38 degrees centigrade. To achieve this objective, we followed these materials and methods. The experiment was conducted in 2017 and 2018 at the SRI Research Farm Udawalawa under these environmental and soil conditions in open top chambers and open field conditions. And 3 factors, including carbon dioxide concentration and air temperatures in treatments, ambient treatment in open field conditions, and ambient and elevated temperature, elevated conditions in open top chambers, and 8 sugarcane varieties as treatments in split plot design with the 5 combination of carbon dioxide and temperature treatments as the main plot treatments, and 8 sugarcane varieties as subplot treatments in 3 replicates, with 4 carbon dioxide temperature treatment combination as ambient and elevated conditions in open top chambers, and 1 ambient carbon dioxide and temperature treatment combination in open field conditions were considered as 5 main treat plots, and 8 sugarcane varieties were considered as subplot treatments. Altogether, these 15 main plots and 240 subplots were allocated into the experiment as shown in the field layout. Basically, we had to construct 12 open top chambers using GI frame and UV treated polythene with the dimension of 3 meter height and 3 meter diameters as shown in the figures. Then after we install all gas cylinders, air blowers, heaters, gas regulators, and air flow regulators. Then after, required carbon dioxide concentration was maintained by injecting pure carbon dioxide from gas cylinders through air blowers and gas regulators, and required air temperature was maintained by setting temperature at 35 degrees centigrade in every morning, and heaters and air blowers automatically switch off when temperature exceeding 35 degrees centigrade. Then after, single budded post seed sets of each variety were planted in each subplot, and experiment was maintained with SRI recommendation with all accessories to measure soil moisture and temperature condition. Then after, soil moisture, soil temperature, air carbon dioxide concentration, and RH were monitored using these sensors and instruments installed inside and outside of the open top chambers. Then after, germination count, tiller count, plant height were measured as growth measurements, and also 1 bush in each subplot uprooted and sampled for estimating the total biomass in stock and bush. And then finally, experiment was harvested to collect final data and obtain number of stocks and the weight of all stocks in each subplot to estimate the biomass accumulation of sugarcane. And then biochemical analysis of cane juice were done at the laboratory of the SRI to calculate sucrose content in cane juice with as the POL and POC's percentage values. Then after, all count data were analyzed using CAD mode, and parametric data were analyzed using ANOVA. Means were separated by using Duncan new multiple range test, and the treatment effect of carbon dioxide and temperature effect on all crop variables were estimated by independent contrast. The carbon dioxide effect was estimated as the difference between elevated carbon dioxide and ambient carbon dioxide in the open top chambers, and elevated temperature effect was estimated as the difference between elevated temperature treatment and ambient temperature treatment in open top chambers, and also combined effect of elevated temperature and carbon dioxide estimated as the difference between the elevation of carbon dioxide and temperature treatment and ambient temperature and ambient carbon dioxide treatment in open top chambers. And also, chamber effect was estimated as the differences between the treatment in open top chambers and the open field conditions, and varieties were compared using the DNMRT values. Then let's see the results and discussion of the findings, what we observed in our experiment. the chambers which had elevated carbon dioxide and elevated temperature combination increased their carbon dioxide concentration by 435 ppm compared to the ambient treatment in open top chambers, and also the chambers which had elevated carbon dioxide, elevated temperature, and both elevation of carbon dioxide and temperature increased their temperature by 0.7, 1.72, and 3.46 degrees centigrade compared to the ambient treatment in open top chambers. And also ambient treatment in open top chambers increased their temperature by 1.63 degrees centigrade compared to the open field condition due to the chamber effect. Then let's see how crop was performed under these climatic condition what we simulated. And basically initially number of shoots per bush showed significant treatment, varietal and treatment into varietal interaction effect, and elevated carbon dioxide increased number of shoots per bush in 2 varieties, and elevated temperature increased number of shoots per bush in 5 varieties. And also combination of elevated carbon dioxide and elevated temperature increased number of shoots per bush in 3 varieties and decrease in 1 varieties. And then after biomass per individual stock showed treatment effects and elevated carbon dioxide increased biomass per stock at 156 days after planting. However, elevated temperature and combination of elevation of carbon dioxide and temperature decreased biomass per stock at the end of the growing season. And also biomass per stock did not show any treatment into variety interaction effect. However, it shows significant genotypic variation, and SL77.5 and 86 nodes is recorded higher biomass at end of the growing season, and SL96 to 37 recorded the lowest biomass of individual stock. However, biomass per bush did not show any significant treatment effects, but it showed the significant genotypic variation. CO775 and SL86 now 1 to 8 recorded higher biomass per bush, and SL88116, which had higher pull and POCS value, recorded the lowest biomass per bush. Then let's see how climate change affects sucrose accumulation of sugarcane. It showed significant treatment, varietal and treatment into varietal interaction effect, and elevated carbon dioxide decreased pollen and POCS in 1 variety. Elevated temperature decreased pollen in 3 varieties and POCS in 2 varieties. And also combination of elevated carbon dioxide and temperature decreased pollen in 3 varieties and 1 in POCS in 1 variety. Those are the results what we observed. Based on these results and discussion, we are coming to this conclusion. Elevated carbon dioxide alone does not influence biomass and sucrose production. Elevated temperature alone and combination of elevated carbon dioxide and temperature do not influence biomass production, but they increase number of shoots in the bush and also increase sucrose accumulation in 5 out of 8 varieties. And also all measured and analyzed variables show significant genotypic variations. Higher values of pollen and POCS in the variety SL88116 are not affected by the elevation of carbon dioxide to temperature individually or combination. Therefore, the variety SL8811 is a valuable genetic source to develop new sugarcane varieties with high sugar content in a future warmer climate. With this information and findings and conclusion, I would like to conclude my presentation with the acknowledgement of this personnel who helped me to conclude and conduct this experiment very successfully. Finally, I would thank all of you for attention me. Thank you very much.
Thank you very much, Mr. Silva. Now the presentation is open for discussion. Yeah, we have one question from Dr. Nalaka.
Thank you, Mr. Silva, for your presentation of very impressive and laborious field work. Has come to a very good end. One question I have is actually, so some of the cultivars shows a reduction in sucrose accumulation. according to your simulations, how would you try to explain this reduction of sucrose accumulation?
Yes, sir. It is a very good question, and also it is the main finding of our research experiment, which is the effect of sucrose accumulation. It is the very remarkable finding, as I showed that biomass production is not affected, even though increasing temperature and increasing both carbon dioxide and temperature increases the number of shoots and decreases individual stock weight. So we hope to increase population biomass, but we expect a reduction of biomass production, because in this experiment we observe that the photosynthesis of majority of sugarcane varieties, especially which showed the reduction of biomass accumulation, and especially you asked the question reduction of sucrose contents, did not respond to the elevation of temperature as well as elevation of carbon dioxide. Then they maintain the similar biomass production, carbon photosynthesis rates under elevated conditions, same as the ambient conditions. But when increasing the temperature, we know that most of literatures say that increasing temperature increase respiration. Then the availability of photosynthetics or carbohydrates to partition into the biomass and sucrose is reduced. That is the main reason of reduction of sucrose accumulation into the stocks in some specific varieties. Those it is a genetically dependent character, and I think their temperature is supra-optimal temperature. Therefore, it affects increase in temperature and reduce the sucrose accumulation into this stock, because mainly the increasing of temperature and carbon dioxide did not increase the photosynthesis as well as the biomass production. That is the main explanation for your question. Is it enough, sir?
Yeah, thank you for the explanation, and that would have been if you had the opportunity to measure respiration as well.
Okay, time is up for the presentation. I thank Mr. De Silva for excellent presentation. Let's move into the next presentation. The title of the next presentation is, that's on fisheries, effect of exotic tilapia on the population of native fish, climbing perch in Malwatuoya Nachaduwa floodplains. The authors are PACT Pereira, CMB Demataveva, TV Sudar Bharati, SP Kodutuaku, and U Edirisinghe. The presenter is CT Pereira.
Sri Lanka is rich in freshwater fish. Nevertheless, 74% of endemic species are threatened with extinction. Exotic fish species are a group of animals that expose native fish species to new competitors. Among exotic fish, tilapia is notorious because tilapia is considered more challenging to native fish. So, are these conclusions reasonable? Ladies and gentlemen, I am PACT Pereira. I would like to share with you my interesting research findings, effect of exotic tilapia on climbing perch, Anabas testudineus in Malwatuoya Nachaduwa floodplain. Malwatuoya is the second longest river in Sri Lanka, and it is a seasonal river and it is mainly fed by northeast monsoon rains. Mallathwaya meanders from Reticula to coast of Mannar across north central and northern provinces of Sri Lanka. So Upper Mallathwaya is the upper part of Mallathwaya is called Upper Mallathwaya. So the Nachaduwa perennial reservoir has been constructed across Upper Mallathwaya. So approximately 0.5 kilometers from Mallathwaya Nachaduwa Reservoir Inlet, there is a large flood valley. During the monsoon rains, Mallathwaya overflows and floods the valley, creating large number of water pockets. However, during the dry season, water disappears from the valley, but leaving large number of isolated water pockets. Many fish species cohabit in these water pockets during different monsoon seasons of the year. So I conducted my research in this water pocket environment in the floodplain. So Nachaduwa perennial reservoir is routinely stocked with exotic fish, and they can invade Upper Mallathwaya and through Mallathwaya Nachaduwa Reservoir Inlet. So the many fish species cohabit in this Mallathwaya Nachaduwa Reservoir Inlet area, and the most abundant species is Anabas testudineus. So they breed in this environment, and therefore their eggs and fry may be under threat. So this is Nachaduwa Reservoir and this is Mallathwaya, and these are the water pockets in the flood valley. So this is how fish enters from Nachaduwa Reservoir into Upper Mallathwaya. So the main objective of this study is to identify the fish behavior and the relative abundance and species, spatial distribution of fish species in water pockets. Other than this, gut content analysis was carried out in fish species found in water pockets. Other than this, as a comparative study, I also conducted gut content analysis for the same species found in Mallathwaya and Nachaduwa Reservoir in order to identify the impacts and compare the impacts in different aquatic environments. So the data were collected by interviewing fishers, observing fish, and also by seining. When seining, I completely water pockets to identify the species richness, relative abundance, and density. Other than this, fry and eggs were also collected. So by completely seining the water pockets, I analyzed gut content of 6 fish species. So this is the map of the study area. It is a 3 kilometer long segment in the flood valley with 5 study sites, and these brown circles indicate the study sites, and approximately there is a 0.5 gap between 2 study sites. Results and discussion. Then composition of the fish species during the dry season. Only 2 species existed during the dry season, and Anabas testudineus dominated the environment. However, during the first intermonsoon season, the highest abundance of Anabas testudineus and 2 Oreochromis species can be found. First intermonsoon season coincided with the breeding season of Anabas testudineus, and highest occurrence of Oreochromis species was due to availability of more eggs and fry. So during the second intermonsoon season, the highest species richness can be found. So the Etroplus and the Devario malabaricus were the most common species, and during the northeast monsoon season, water level of the flood valley was high. Therefore, the highest relative abundance of the Etroplus sudatensis in this environment was due to availability of more space and favorable water quality. So when we consider the spatial distribution, these graphs clearly show that both Anabas testudineus and Channa striata, or the density of both Oreochromis species and Channa striata, had decreased towards the upstream side of the valley. So therefore, there is a significant positive correlation between the density of Anabas testudineus and Channa strata. So therefore, there are little impacts from one species on the survival of other species. However, the situation is completely different during the first intermonsoon season. Sorry. Density of Anabas testudineus has increased toward the upstream side, while the density of Oreochromis species has decreased toward the upstream side. Therefore, during the first intermonsoon season, there is a significant negative linear relationship between the density of Anabas testudineus and density of Oreochromis mozambicus and Oreochromis niloticus. Therefore, there are impacts from one species on the survival of other species. So this graph shows that during the second intermonsoon season and the northeast monsoon season, the similar pattern of variation in their distribution can be found. Therefore, during the first intermonsoon season, second intermonsoon season, and the northeast monsoon season, there are impacts from one species on the survival of other species. However, when we want to look into this matter more little bit closer. So these graphs can explain that during the first intermonsoon season, there is a significant negative linear relationship between the density of Oreocomis niloticus and density of fry of Anabas testudineus. Similarly, there is a significant negative linear relationship between the density of Oreocomis mozambicus and fry density of Anabas testudineus fry. Similar relationship can be found during the second intermonsoon season as well. Therefore, it is clear that there are impacts from Oreocomis species on the survival of fry of Anabas testudineus. However, sorry, there is no significant relationship between Oreocomis species and the fry density of Anabas testudineus during the northeast monsoon season. This is mainly due to higher water level in the flood valley. When we consider the eggs of Anabas testudineus, there is a significant negative linear relationship between the density of Oreocomis niloticus and egg density of Anabas testudineus during the first intermonsoon season. Also, there is a significant negative linear relationship between the egg density of Oreocomis niloticus and Oreocomis mozambicus and egg density of Anabas testudineus. Similar relationship can be found during the second intermonsoon season as well. Therefore, it is clear there are impacts from both Oreocomis species on the survival of eggs and fry of Anabas testudineus. When we consider this matter more closely, it is important to identify or to study the gut content results. So this graph shows that there is an Oreocomis niloticus has ingested 24% of fish fry during the first intermonsoon season, and Oreocomis mozambicus has ingested 22% of fish fry during the first intermonsoon season. During the second intermonsoon season, niloticus has ingested 22% of fish fry and mozambicus has ingested 11% of fish fry. Therefore, it is clear, other than this, they have ingested considerable number of fish eggs as well. Therefore, in this flood-created environment, these two Oreocomis species are more predaceous. However, when we consider the northeast monsoon season, it is clear that the amount of fry and eggs in the gut content of both Oreocomis species was low, and however, their gut contained more content of animal matter. This is because actually when they are in difficulty of finding eggs and fry, they try to eat more plant matter. This shows their flexibility in their feeding habits. When we compare the gut content results of two Oreocomis species with the highly carnivorous species Channa striata has ingested 10% of fish fry during the first intermonsoon season, but both Oreocomis species has ingested considerably higher number of fish fry. During the second intermonsoon season, Channa has ingested only 20% of fish fry, but both Oreocomis species has ingested considerably higher number of eggs and fry. Therefore, in this flood-created environment, both Oreocomis species are more predaceous than highly carnivorous Channa striata. When we compare the gut content results of Anabas testudineus with Oreocomis species, Anabas has ingested only 6% of fish fry, but both Oreocomis species has ingested considerably higher number of fish fry during the first intermonsoon season. During the second intermonsoon season also, Anabas has ingested only 4% of fish fry, but both Oreocomis species has ingested a considerable higher number of fish fry and eggs. Therefore, in this flood-created environment, these both Oreocomis species are more predaceous than Anabas testudineus. However, Heteroplus suratensis has not ingested fish fry or eggs. Therefore, it is clear of all the fishes, highest quantity of eggs and fry have been ingested by Oreocomis species, and the highest predation is by Oreocomis niloticus. So when we compare the gut content results in Upper Mallathura, once again, the highest predation is by Channa striata and the second highest predation is by Anabas testudineus. However, the amount of fry and eggs ingested by both Oreocomis species is at a low level. When we consider the results of the gut content results of Nachyaduvarisovale, once again the highest predation is by Channa striata. However, the amount of fry and eggs found in the gut content of Anabas studioneosum to Oreocomis species is more or less similar. So when we compare these three aquatic environments, therefore it is clear the highest predation is in flood-created environment in water pockets, and the lowest predation is in upper Mallathuwe natural riverine habitats. So this is actually the similar situation can be found during the second intermonsoon and northeast monsoon seasons. Therefore, it can be concluded that there are no significant impacts due to Atropos suritensis in this flood-created environment, but Channa's data showed significant impacts in flood-created environment, in the man-made environment, and in the riverine environment, nevertheless show low impacts compared with Oreocomis species. However, Oreocomis species predate on eggs and fry, and Oreocomis niloticus showed comparatively higher impacts than Oreocomis mozambicus. So therefore, the predatory behavior is high in flood-created environment as there is no protective habitat for fish, eggs, and fry. Therefore, the current study does not oversimplify the feeding behavior of Oreocomis species and does not label Oreocomis species as a noxious group of fish that are equally deleterious in every aquatic environment in Sri Lanka. Actually, the impact of Oreocomis species on native species depend on the type of the environment they inhabit. Therefore, I would like to conclude this presentation with this take-home message. If we can protect our natural environment, we can mitigate the exotic impacts. So thank you very much for your kind attention.
Thank you very much, Mr. Pereira, for an excellent and elaborative presentation. And now the presentation is open for discussion. We didn't see any responses, but eventually we will. Dr. Nalaka is there. Yes, Dr. Nalaka.
Thank you, sir. Yes, thank you, Mr. Pereira, for your very nice presentation. My question is to further clarify the statement you made at the last part of the presentation. So, if I understood well, Malvathuwe segment has low impact from these exotic species. Is it primarily because the Malvathuwe is not a prominent ground for laying eggs and hatching their fry, or is it because of a particular behavioral differences of this exotic species? Your microphone is mute.
Okay, the reason is actually when we compare these three aquatic environments, we can find the large number of natural habitats or the cohabiting places for fish in natural riverine habitats. It may be leaf logs and stones and riverine habitats and river banks and roots and so many things. But in the flood-created environment, it is a bare environment with only grass. So when fish, the Oreocomis, not the Oreocomis species, but our native species have enough cohabiting places in natural riverine habitats. So actually, there is a problem to find their eggs and fry for these Oreocomis species. So they have very good protected environment in the riverine habitats. So very special thing is when we compare this with the Nachaduva reservoir. So Nachaduva reservoir is a man-made reservoir. So actually in Nachaduva reservoir, I didn't find any natural habitat like riverine environment in Mallathuwe. So when we compare this Mallathuwe environment with the Nachaduva environment, that is why the impact is high, because they have very less we can say number of cohabiting places for fish for their protection of fry and egg especially. But in the flood-treated environment, there is no protection at all. That is why origami species have chances. Although we say origami species are herbivores, so when they find exo fry, whatever, they eat when it is available. Therefore, what the best solution for this actually is, if we can protect the riverine cohabitat places of our.
I think your time is up. I have to stop. Let's move on to the next presentation. Thank you very much, Mr. Pereira. That is cellulose decomposition potential of soil as affected by vegetable cultivation. A case study in Cascade district, Sri Lanka. The authors are W.A.M.S. Vikramarachi and W.S. Dandeniya. The presenter is Vikramarachi. It's for you.
Hello, ladies and gentlemen. I am W.A.M.S. Vikramarachi and I am here to present my research findings regarding cellulose decomposition potential of soil as affected by vegetable cultivation, a case study in Cascade district, Sri Lanka. In soil, there are several nutrient cycles like carbon cycle, nitrogen cycle, sulfur cycle, and phosphorus cycle. When we consider all of those cycles, the decomposers are very important. When consider the carbon cycles to decompose soil organic matter, decomposers are mainly involved. Therefore, if a farmer knows their field carbon decomposition potential, he can get an idea about the carbon dioxide emission of their soil and also the carbon storage and short-term nutrient turnover. In soil, according to the different management practices, the carbon sequestration may be changed. In soil, carbon decomposition potential is a complex process. It involves many organisms. There are three methods to determine carbon decomposition potential. One is mass balancing, and the other one is carbon dioxide emission, and finally the tracer technique. In here, this study, we practice a new technique, qualitative method to determine carbon decomposition potential. In this study, we made our objective as this, to assess cellulose decomposition potential of soils as affected by vegetable cultivation. In the methodology, for our study location, we selected Kanangama and Atulagama Gramanidari divisions in Cascade district, Sri Lanka. In there, we selected 10 vegetable fields and 5 sites under natural vegetation. For the 10 vegetable fields, we included 3 organic fertilizer using lands, 3 synthetic fertilizer using lands, and 3 organic plus synthetic fertilizer using lands. In soil sampling, we done in 0 to 50 centimeter depth, and we selected 2 sampling points from each study site. In the laboratory analysis, we analyze soil pH, organic carbon percentage, and active carbon content. This study was practiced in 2 ways. One is in situ and the other one is ex situ. For the in situ study, we prepared litter bags. Litter bags were prepared using nylon mesh. Litter bags were 20 to 7 centimeter in size. We divide our prepared litter bags into 2 groups. For one set of litter bag, we added dried cotton wool, and for the other set, we added 2 filter paper for each litter bag. All of those litter bags were placed our selected sites, and finally, we remove out those litter bags. The cotton wool litter bags was removed out within 6-week period, and within 4-week period, we remove out filter paper included litter bags. After that, in the laboratory, we analyzed the decomposition rate using mass balance technique. We see the in situ experiment. ex situ experiment, it was conducted in the laboratory as an incubation study. In there, we taken 100 grams of soil. It was air dried and saved. It was added 25 milliliter of water and quarter piece of filter paper was added to a petri dish. After that, it was added that prepared soil and the petri dishes were sealed well. After that, it was put in an incubator for 2-week period. And finally, we remove out from the incubator and captured the filter paper using a camera. The image was analyzed using an image software, and according to the results, we taken the digested percentage. The statistical analysis was done using SAS University Edition software. In there, analysis of variation was done using general linear model procedure. The means comparison was done using least significant difference method, and correlation was done using Pearson correlation method. Results and discussion. The characteristic of soil was like this. The pH of the all soil samples were acidic because the soil samples were collected from wet zone Sri Lanka. In wet zone, normally gets high rainfall, therefore the basic cations are removed out from the leach down from the soil. Therefore, the remaining acidic cations create acidity in soil, and normally in wet zone area, soil becomes somewhat acidic. When we consider organic carbon percentage, all the values showed above 3. Therefore, the organic carbon percentage of analyzed soil samples are somewhat high. When consider the uncultivated soil and the organic fertilizer used in soil, it showed about higher amount of organic carbon percentage, and the lowest organic carbon percentage showed organic fertilizer used in lands. And significant difference was showed the organic fertilizer used in land from the other values. When we consider organic active carbon content, it was in normal soil active carbon content range. It was about 274 milligram per kilogram to 359 milligram per kilogram. When consider in situ analysis, when using cotton wool, after 6-week period, the litter bags cotton wool was degraded about 61% to 65%. Therefore, it was very difficult to observe the treatment effect. Therefore, we would like to suggest here, conduct in situ experiment for only 4 weeks. When consider in situ experiment when using filter papers, about 44% of samples exhibited over 80% of decomposition. Therefore, in here also it was very difficult to identify the treatment effect. When consider ex situ analysis results, the carbon decomposition potential was significantly affected by the management practices. It was clearly observed by the graph. When we consider synthetic fertilizer used in land, it showed the higher amount of degradation. The lowest degradation is showed organic fertilizer used in land. If we take the organic carbon percentage of those 3 management practices, the lowest organic carbon percentage was the organic fertilizer used in lands, and the higher shows the organic plus synthetic fertilizer used in lands. So, according to the results, we can say if we added more amount of organic carbon to soil, it will not increase the organic carbon percentage as well as the organic carbon degradation. In soil, there are several decomposers according to the management practices. Those decomposers are differ according to the management practices. We can say R strategic decomposers and K strategic decomposers. Those decomposers, the decomposition rate is different. Thereby, according to the management practices, the decomposition rate amount is different. And finally, we analyze the correlation between ex situ analysis results and in situ analysis results, but those results were not significant. Therefore, we can conclude our study results as this. 75% samples that showed over 80% degradation were from cultivated lands. Vegetable cultivation affected carbon decomposition potential differently depending on the nature of cellulose input and the history of fertilizer management. Although not significant, the result indicated a trend that carbon decomposition potential is increased in soil with cultivation. We can suggest cellulose filter paper digestion assay can be used to assess the effect of management practices on carbon decomposition potential. In situ incubation study with cotton wool should be conducted in less than 6 weeks of duration to observe treatment effect in the studied fields. Conducting farmer participatory research, as in here, would make farmers more aware and concerned about soil fertility aspects and help knowledge dissemination. These are the references I use. And finally, I would like to acknowledge my supervisor, all the demonstrators and non-academic staff, Department of Soil Science, University of Peradeniya, my brother and all the farmers who helped me to conduct the experiment in their fields. Thank you for hearing me.
Thank you very much, Ms. Vikramarach, for an excellent and elaborative presentation. The paper is open for discussion. There is one, Dr. Tusita Amarasekara. Yes.
Okay, Ms. Vikramarach, can you hear me?
Yes, sir, I can.
Yeah. Okay, actually, this is not exactly a question, actually, for my information as well. Now, in our findings, it says that the laboratory incubation, in your laboratory incubation results showed that the soil collected from cultivated lands had higher CDP compared to the uncultivated.
Yes.
Yeah, could you please a little bit explain about what would be the reason for this actually?
Yeah. Yes, sir. Can you explain again? I can't understand.
Yeah, in your incubation test, laboratory test, findings showed that the soil collected from cultivated, cultivated means synthetic fertilizer added.
Yes, sir.
Showed higher CDP compared to the uncultivated. Is that correct?
Yes, sir.
Yeah. What is the reason?
Here, actually, this result is not sufficient for this question, for giving answer for this question. Actually, sir, in soil, there are two types of degradators, like R strategic and K strategic. When consider R strategic decomposers, they are active fast when minerals are high in that soil. Therefore, in synthetic fertilizer using lands, normally added fertilizer and they get frequently minerals, nutrients to soil.
Yeah.
That's why that decomposers are, I think, growing fastly in that soil. But when consider the organic and uncultivated soil, they are not using these synthetic nutrients to soil. That's why the in that soil may be present K strategic decomposers like this, that decomposes. Those decomposers, the rate is low, decomposition rate is low. That may be the results for that.
Okay, Ms. Vikramarach, in integrated nutrient management, normally we recommend to add synthetic fertilizers and organic manures together. Combination of synthetic and organic manures.
Yes,
in terms of CO2 emission, right? I'm asking in terms of CO2 emission, not regarding this the plant nutrition. What do you think about this kind of recommendation? Application of synthetic fertilizer plus organic manure together into the soil?
Actually, sir, if there's use organic plus synthetic fertilizer, in that soil may be present these K strategic as well as R strategic, both of decomposers. Thereby, the carbon dioxide emission may be not more than higher as well as not more than lower. It may be maintained in a middle level. Therefore, it is very suitable for if we apply synthetic plus organic fertilizer both to the farm.
Okay, yeah, thank you very much.
Okay, Dr. Nalaka, you can present your question.
Thank you, Ms. Vikramarachi, for your interesting presentation. Only one question I have. Did you measure or observe any difference in termite activity, whether their presence or their activity in different sites?
Actually, I couldn't find any termites, sir. But I can't. I didn't observe, but I don't know they are present or not. It may be present, sir.
Yeah, better if you are replicating or doing the same experiment again, better to observe those animals or fauna who are involved in decomposition, because
Yes,
they are very likely to eat these
Yes.
filter papers. Thank you.
Yes, sir. Thank you, sir.
I have a quick question. 1 minute, right? You just give an answer yes or no, right? Now you use pure cellulose, right?
Yes, sir.
Right. Usually when we do cultivation, you add complex cellulose as organic matter or plant material, which contains varying levels of lignin, which affect the cellulose degradation, right? Do you think your results would have been different if you use a complex cellulose?
Yes, sir. Actually, it may be.
Okay, thank you very much. Thank you very much, Miss Vikramarachi.
Yes, sir. Actually, it may be different.
Okay, right. Thank you very much. Thank you very much, Ms. Vikramarachi, for an excellent presentation. And let's move on to the next presentation. The title of the presentation is Micronutrient Status in Commercial Sugarcane Growing Alphisols at Sevanagala, Sri Lanka. The authors are H.A.S. Veerasinghe, G.P. Gunaratne, A.N. Jayakodi, and R.S. Dharmakirti. The presenter is H.A.S. Veerasinghe. Please start your presentation.
I'm going to present a component of my PhD research study, which was carried out under the Postgraduate Institute of Agriculture. My title is Micronutrient Status in Commercial Sugarcane Growing Alphisols at Sevanagala, Sri Lanka. Sugarcane is an important component to produce sugar locally. And at present, due to the low sugar production in the country, there's a high price fluctuation which has affected the people, and it has become a very important commodity at present. Let's move on to see the current sugar status in Sri Lanka. It has a very high per capita consumption, that is 30 kilograms per year per person, and that accounts to a sugar requirement around 650,000 metric tons annually. The local sugar production is around 9%, which is produced at Palwatha, Sevanagala, Hurana, and Athimale plantations. The balance 91% is imported to the country at a huge cost, around Sri Lankan rupees 35 billion. So let's move on to see the importance of sugarcane crop and the background behind it before the crop. It's a C4 plant. It's annually harvested. It stores sugars in the stalk, and it's bearing a true grass and efficient photosynthesizer. It's well adapted to the dry zone and climate change. It has a minimum water requirement around 800 to 1500 millimeters. So therefore, it is well adapted to the dry zone in the country. So the main sugarcane growing areas are in Kantale, Higurana, Athimale, Palwatha. The next is Sevenagala, which I have selected for the study. The Sevenagala component includes Walave Mahagallar, Ketagallar, Sevenagala Association. And the Walave Mahagallar soils are reddish brown earth soils, simply it's RB soils. The Ketagallar soils are denoted as low humid clay soils and simply denoted as LHG soils. Taking a landscape or the profile landscape around the area, the reddish brown earth soils account to about 90 to 95% in the area study site, and it occupies the upper catina of the landscape. And the lower catina is occupied by the low humid clay soils, which accounts to about 5%. And that's an average slope in the area of 1.5%. The average yield is very important for the crop. Under irrigated cultivation, the average yield is around 100 tons per hectare. Under rain-fed cultivation, the average yield is around 60 tons per hectare. The commercial value for crushing 1 ton of sugarcane at Sevenagala is around ₹17,000. So this accounts that sugarcane is a very important crop and a very productive crop for the dry zone and marginal lands. Let's move on to see the nutrient absorption of sugarcane. It's a heavy feeder of nutrients. It uses to produce 100 tons per hectare. To produce 100 tons per hectare, apart from the macronutrients, micronutrients are absorbed. Ferrous, manganese, zinc, and copper are highlighted, and they absorb at considerably high levels. So what are micronutrients? They are an essential group of plant nutrients that are found in trace amounts in tissues, but play an imperative role in plant growth and development to increase the crop production. So what are the micronutrients available? Ferrous, manganese, zinc, copper, boron, molybdenum, and chlorine. So most discussed micronutrients for sugarcane are ferrous, manganese, zinc, and copper. How soils could be deficient in micronutrients? This is an important component because sugarcane is a long-term cultivated monoculture at Sevenagala. It's almost around 35 years. So this monoculture has or could be an important factor that has reduced the soil nutrients. So continuous use of fertilizer with low levels of micronutrients, because that is the case with present at Sevenagala. So increased agricultural production. Agricultural production is increased. Depletion of soil organic matter, which helps the soil to retain more nutrients, and increase cultivation in the areas with low fertility. With that, the objectives of this study is to expose the micronutrient status of sugarcane growing soils at Sevenagala and to identify the potential micronutrients that could be deficient. Moving on to the materials and methods. The study site was selected, as mentioned earlier, Sevenagala, and it has rain-fed cropping systems, irrigated cropping systems. Then under both cropping systems, there are low humid clay soils and reddish brown earth soils. And there are 1 to 6 divisions, management divisions at the area, and is given in this diagram. Soil sampling. It was carried out under the 6 management divisions, and 263 soil samples were collected from the 0 to 15 soil centimeter depth. Under the 6 management divisions, 122 samples were collected from the rain-fed cropping system, and 141 samples was collected from the irrigated cropping system. And from both cropping systems, there were 208 soil samples under reddish brown earth soils and 55 soil samples which came upon low humid clay soils. Once the samples were collected from the field, they were brought to the laboratory at the Crop Nutrition Division of the Sugarcane Research Institute, and they were air dried, saved, and seed soil samples were collected and extracted to measure the ferrous, manganese, zinc, and copper using the DTPA extraction. The data was analyzed and statistically analyzed as well. The data was normally distributed. Analysis of variance was carried out using PropGML procedure of SAS. Moving on to the results and discussion part. The results summary shows that all ferrous, manganese, zinc, copper has a very high coefficient of variance. So it shows that there's a wide variation within the sugarcane growing area at 711. And considering individual components, ferrous has a minimum around 7.4 and the maximum is around 342. The manganese was at very low concentrations and moved up to 1162, which was the maximum. Zinc it was at very low levels and it ranged up to 4 milligrams per kilogram, and copper it ranged from very low levels up to 23. Moving on to the individual elements, the soil available ferrous. It was significantly high under rain-fed cropping system compared to the irrigated cropping system at 711. And then available ferrous was significantly high under rich brown earth soils compared to low humic clay soils. Considering soil available manganese, there was no significant difference under the main cropping systems and the soil types. That is because of the variation. And available zinc, there was a significant difference between it was significantly high under reddish brown earth soils compared to the low humic clay soils, considering the soil types, but there was no significant difference on the main cropping systems. Considering the available soil copper, it was significantly high under reddish brown earth soils compared to the low humic clay soils at 711. The main cropping systems at 711, there was no significant difference. Considering all these results, it was summarized and the micronutrient deficient percentages of sample sites were identified. For this, there were 3 classification systems were used, and one is Poach and Hunter 2002, then Verma and in 2004 and Calcino in 2010. The latter 2 were considered as the classification systems for specially designed for sugarcane crop. And this was compared and it identified that zinc showed deficient patches in considering all 3 classification systems, and zinc has a serious problem at the considered study site. And apart from that, there's concerns should be attended to copper as well. With that, the micronutrient availability in sugarcane growing soils at 7 are significantly affected by the cropping systems, that is the rain-fed and irrigated cropping system, and the soil types, reddish brown earth soils and low humic clay soils. The reference classification provide a base to identify percentages of sample sites that are deficient for the micronutrient for optimal crop growth. The ferrous and manganese were comparatively at sufficient levels for sugarcane, and zinc and copper deficient patches are at present in all cropping systems and soil types. So it's very important to consider more on zinc because this affects the sugar quality of the cane stock, and therefore attention is required. So even though there are no plant deficient symptoms in the field, precaution measures for micronutrients are required to mitigate the negative effects on the crop yield and quality in the near future. And with that, the importance to the sugar industry is the mapping the key soil micronutrients across the sugarcane growing area at 7 and provide information for the sugar industry with solutions for deficient micronutrients. And with that, I conclude my presentation. I thank you very much for your attention. Thank you.
Thank you very much, Mr. Veerasinghe, for an excellent and elaborative presentation. Now the floor is open for discussion. Dr. Tusita has raised the hand. Yes, Dr. Tusita.
Okay, Mr. Veerasinghe?
Yes, sir. Yes, sir.
Okay. Okay, thank you very much for a very interesting presentation. I have one actual question. According to your findings, copper and zinc are deficient, and when it comes to the LHG, the problem is more severe. Is that?
Yes, sir.
Yeah. What? Yeah, please.
According to the information, zinc was the most important and deficient component what I got with my results.
Yeah. Now, when you compare to reddish brown earth and LHG, the problem is more severe in LHG, no?
Yes.
Yeah. What would be the reason for that?
According to the information what I have, data what I have collected, LHG, there are more cations, calcium, magnesium, and sodium cations that have been accumulated, and that develops the salinity. And I assume that because of those cationic adherence, there is low availability for zinc and other things to be retained in the soil.
So what about soil pH? Have you measured soil pH in both RB and LHG?
Yes, I have measured.
Yeah. Is there any relationship between soil pH and this nutrient availability? Have you found any relationship?
Yes, soil pH in reddish brown earth soils range from around 5.5 to 7.5. But when it goes to LHG soils, the soil pH goes up to 9. So soil pH is also one factor for the availability in the nutrient, micronutrient availability.
It may be a one main factor.
Yeah.
Okay. Okay, thank you very much. Thank you.
Okay, thank you, sir.
Yes, I have a small question in the same line.
Yes, sir.
We know at the low pH, the micronutrients what you have mentioned now become more available. And when it goes to higher pH, it becomes restricted, right?
Yes, sir.
Right.
Especially in the LHG soils.
Yes. So, do you measure the available micronutrients, especially zinc and copper, or the total?
I have presented the available micronutrients in this study.
Available, soluble, right?
Yeah.
Not the total. Not the total.
No, total I have measured, but I have not included here because it will get complicated.
Is there a difference, big difference between the total and the available?
Yes, sir. The total amount is much higher. Only the available is available component is much less.
Now, if you take the sufficient level of the plant, sugarcane,
yes.
Right? We know the requirement of sufficient level.
Yes.
So the available micronutrients in especially copper and zinc, are they high in total amount?
Yeah, they are high in total amounts.
Right. That
only
means,
the available
that means it may be mainly due to, as Dr. Tusita mentioned, maybe due to the pH of the soil.
pH of the soil could be one reason, sir.
Yes. I think the time is over. So thank you very much, Mr. Veerasinghe, for excellent and very comprehensive presentation. It's very interesting. Let's move on to the final paper of the day. The title of the paper is Analysis of Water Productivity in Paddy Cultivation within the Ulagalla Tank Cascade System in Sri Lanka. The authors are KKPN Kahatuduwa and DVP Prasad. The presenter is PN Kahatuduwa. Please present your paper.
Hello, and I'm Pimika Kahatuduwa. The research study that I'm going to present is an analysis of water productivity in paddy cultivation within the Ulagalla Tank Cascade system in Sri Lanka. This is the outline of the presentation. Let me start with a brief introduction. According to Madhuru Bandara, the tank cascade system in Sri Lanka is a connected series of tanks organized within the micro-catchment of the dry zone landscape for storing, conveying, and utilizing water from an ephemeral river. Surplus water from upstream tanks and return flows from rice irrigation are captured in downstream tanks and are thus used in the command area of second tank. E.T. and again Pushpamar have stated that the major reason for the formation of tank cascade systems in the dry zone of the Sri Lanka is the temporal and spatial disparities of annual rainfall, which highlighted the need for a proper water management. Here is an illustration of a typical land tank cascade system. The selected research site for the study was Ulagala tank cascade system. The function and productivity of Ulagala tank cascade system are mostly unknown, as a very few research studies are carried out regarding this research site. Ulagala tank cascade is located in Tirupani and Kekirawa divisional secretariats in the dry zone of Sri Lanka, adjacent to Tirupani and Mahapanamula tank cascades. The cascade covers around 51 square kilometers of land, originally with 19 completely rain-fed tanks. Of these tanks, only 9 tanks are functioning as irrigational tanks at the moment. The two main research objectives identified for this study are: first one, to estimate the hydrological endowment of the Ulagala tank cascade system. Second one, to explore the variation and the determinants of upstream and downstream water productivity of Ulagala tank cascade system. Moving on to methodology. 9 tanks in the cascade belong to Tirupani and Kekirawa divisional secretariats were studied in this study. Both primary and secondary data for the study were collected through field surveys and household surveys. A household survey was carried out from February to March 2020. Samples were selected to cover 25% of farmer organizations' memberships. This includes 107 farm households. Cross-sectional survey methodology was used for this study. A comparative study was carried out to compare upstream and downstream, as well as Yala and Maha seasons. Based on that, 4 scenarios was classified as upstream Yala, upstream Maha, downstream Yala, and downstream Maha. The boundary of the upstream and downstream of the cascade were demarcated at the point where the cumulative water surface areas of upstream and downstream tanks were approximately equal. Field level water productivity was calculated using the given equation in current slide. The empirical model used for the study is shown in the current slide. Moving into results and discussions. The hydrological endowment is an important parameter for identifying cascades, functions, and performances, which is an indication of the adequacy or the inadequacy of water for feeding the existing feeding area. In Ulagala Cascade, the ratio total catchment area of the cascade to the total water spread area of the all tank located within the cascade is 8.74, and the ratio of the total command area under all small tank to the total water spread area of all tanks located within the cascade is 0.97. Technically, according to the above values, the cascade is in a state where it can feed its command area for both Yala and Maha seasons. However, almost all the tanks in the cascade are silted. Hence, the water holding capacity of Ulagala Cascade as a whole has reduced approximately by half of its initial capacity. In the illustration, it is shown how the average effective depth of the tanks have changed over 20 years of period. In the current slide, the descriptive statistics for the study are shown. The specific points will be discussed in the next slide. The highest paddy yield is recorded in the Maha season of the upstream, but paddy yields in all four scenarios were lower than the average paddy yield values for rain-fed systems in the Anuradhapura district for respective year. Comparing with the regional benchmarks for the water productivity in paddy, the paddy water productivity balance in Ulagala Cascade are in a satisfactory levels for upstream Yala, upstream Maha, and downstream Maha scenarios. In the Yala season of year 2019, the cultivated area was considerably lower than Maha in both upstream and downstream, as the farmers are reluctant to cultivate the paddy lands in the Yala season since the occurrence of rainfall and the amount of rainfall are uncertain. The amount of average fertilizer used across the Cascade wells higher than the chemical fertilizer subsidy given by the government. The current slide gives the regression analysis for the study. The specific points will be discussed in the next slide. Wildlife conflicts, previous crop losses due to natural disasters had significant negative relationships with the paddy, water productivity in paddy cultivation. The main wildlife threats were reported as the elephants and peacocks, whereas command area had significant positive relationship with water productivity in paddy cultivation in Yala season of both upstream and downstream. Tank water Hemi had a significant positive relationship with water productivity in paddy cultivation in Yala and Maha season of both upstream and downstream. Availability of lining in the field canal had significant positive relationship with water productivity in paddy cultivation in the Maha season of both upstream and downstream. Moving on into the conclusion. The water productivity in paddy cultivation in Ulagala Cascade is in a satisfactory level compared to the regional benchmarks. However, the increment of tank infrastructure, farmer knowledge on water management, and novel remedies for human-elephant conflicts would make the water productivity in paddy of Ulagala Cascade even better. In order to achieve this, more research on tank cascade systems and policy reforms on tank rehabilitation, infrastructure development, water management, farm education, resolving wildlife conflicts are needed. Here are the references that were used for this presentation. Thank you. Have a nice day.
Thank you very much, Ms. Kahatudua, for an excellent and elaborative presentation. Now the paper is open for discussion. Yes, Dr. Tusita.
Ms. Kahatudua, I think you can hear me.
Yes, sir.
Okay, very good presentation.
Can you come on the screen, please? Ms. Kahatudua?
So I have.
Okay, right. Okay.
Right. So according to your findings, the water productivity of this Ulagala Cascade is in a satisfactory level.
Yes, sir.
You know, the present policy change of the country, that is, you know, go more towards, you know, organic farming, right?
Yes, sir.
Zero chemical fertilizer application.
Yes, sir.
How does it affect this policy on the productivity of the cascade in according to your findings as well as your knowledge?
So according to my point of view, we have to do more research on this issue, because we have as a country we have used this inorganic fertilizer for paddy cultivation for a long period. So we have to have better idea of the sites and the about these policies. But I think, according to my point of view, I think we have to have more research, and then we have to come up with these policies. And according to what I have observed during my field visits, these farmers are addicted, have adapted to these inorganic fertilizers. So more rather than having policies on this, we have to go to the farmer level and discuss with them, and we have to get their ideas also, and we have to do more more changes in their perspectives, I guess. So.
Okay, thank you very much, Ms. Kahatudua.
Okay, and now Dr. Professor Maujud.
Yes, thank you very much for the presentation. I have just simple questions. How you demarcate the upstream and the downstream in the tank cascade systems and calculated the productivity?
Sir, we have set the boundary of the upstream and downstream of the cascade, where the point where the accumulated water surface area of upstream and downstream were approximately equal, sir.
Normally, normally then we have a tank, and in the downstream you have these paddy fields, right? So tank has a catchment, and the down you have a paddy field.
Yes.
So how in the paddy field did you demarcate upstream paddy field and the downstream paddy field, or how you made that productivity different?
Sir, as a whole, we took the cascade as a whole, then demarcated the upstream and downstream, sir, not the paddy tract, sir.
Yeah. Thank you.
Thank you, sir.
Okay, Doctor, I think I'll make a small comment.
Yes,
Dr. Tusita made a comment on application of organic fertilizer in paddy fields. Right, Dr. Tusita? Yes.
Yeah.
I think when we talk about through that we cultivated our rice on organic fertilizer, some our forefathers, true. But you also have to remember, when they use organic fertilizer, they never use compost. They use green manure. They never make compost and add it to the soil.
Yeah, that's right. Yes.
Right?
Yeah.
They add green manure. The only thing they compost or allowed to mature was the animal manure.
Yeah.
Right?
Yeah.
Only the animal manure. The rest is green manure. So I don't know why the people, present day scientists are confusing with paddy cultivation, confusing with organic manure and the green manure.
Yes.
They selected the green manure. This for your information. Selected the green manure, which have higher lignification in their material. So they decompose in different times.
Yeah.
Like we put T1, TD1, TD2, TD3, like top dressings. They mature over a period of time. They decompose over a period of time, like a top dressing, right? So I think the soil scientist and the agronomist, when you talk about organic matter application to the soil, or especially paddy, I think they have to consider these are ancient techniques, how they used it. In our old agricultural texts, they don't talk much about organic manure, but they talk mostly on green manure.
That's
Thank
right.
you.
Green manure plus the
animal manure, yes.
Animal manure, yes. Yeah.
Since there are no more questions, I have to close the session. First of all, I want to, thank you, Ms. Kahatudua, for excellent and very elaborative, comprehensive presentation. So let me take this opportunity to thank all the presenters for their very well prepared and organized and elaborative, very comprehensive presentations, and all the judges who helped and asking questions and even to help evaluate the presenters, and all participants, presenters, judges, and the others, and the coordinator of this session, and Miss Madhushan for recording, and the other staff of the PGIA, and also the PGIA given me the opportunity to chair this session. And thank you very much. You all have a good day and stay safe and God bless you. The session is closed.
All right, thank you very much, sir. Yeah, now all the presentations are over. So let me thank you all. First, thank you very much, sir, the chair, Professor Nimal Pereira, sir, for chairing the session and conducting the discussion in the most fruitful way. We also have a small token of appreciation for you. Thank you, sir. And also let me thank all the panel of judges, Professor Cruz, Dr. Gikeneke, and Professor Amarsekara. Thank you very much, sirs, for your valuable time. And also we kindly request you to complete the evaluation sheets as soon as possible, since we have to analyze the marks and announce the winners in the afternoon. And also a very big thank you to all the presenters for your valuable contribution for this session. So the technical session 3 is now completed. However, we have a small video clip about the boards of studies of the PGIA, and afterwards we are going to show you a couple of slides about the afternoon's schedule. So please remain with us, and also please join the afternoon session as well. Thank you again.